Assalamualaikum and hello everyone. My name is Jana. I will be presenting a CME on Physiology of Hearing. This is the outline of my presentation. I will explain a little bit about sound, about ear and its part and functions, and then auditory transduction, cochlear and organ of Corti, the process of encoding of sound, auditory pathway and auditory cortex. So firstly, what is sound? It is a form of energy produced by a vibrating object. Sound waves are the pattern of disturbances caused by the movement of the energy traveling through a medium as it propagates away from the source of the sound. Sound wave consists of compression and refraction of molecules of the medium in which it travels. Then the velocity of sound is different in different media. The highest velocity is in solid, while the lowest velocity of sound is in air. What is hearing? It is auditory perception and also known as the ability to perceive sounds through an organ, such as an ear, by detecting vibrations as periodic changes in the pressure of surrounding medium. And sound is measured in units called decibels, and the higher the decibel level, the louder the noise. So what is decibel? It is 1/10 of the bell. It is not an absolute figure, but represents a logarithm ratio between two sounds, the sound being described and the reference sound. So actually, the decibel notation was introduced to avoid dealing with large figures of sound pressure level. So sound in decibel means 10 log power of sound 1 over power of sound 0. The sound 1 being the sound being described, while the sound 0 being the reference sound. So at a distance of 1 meter, intensity of a whisper is equivalent to 30 decibel. Normal conversation is about 60 decibel. Shout 90 decibel. Discomfort of the ear around 120 decibel. And 130 decibel will cause pain in the ear. So these are some of the terms that we need to know to understand the physiology of hearing. Firstly, frequency. Frequency is the number of cycles per second. The unit is hertz. 1000 hertz means 1000 cycles per second. Pitch. It is sensation produced by frequency of sound. Higher the frequency, greater is the pitch. Pure tone. It is a single frequency sound, and in pure tone audiometry, we measure the threshold of hearing in decibels for various pure tone from 125 Hz to 8K Hz. Complex sound is a sound that contains more than one frequency, and an example of complex sound is human voice. Intensity. It is the strength of sound which determines its loudness. It is measured by decibels. Loudness, it is sensation produced by intensity. The more the intensity of sound, the greater the loudness. This is audiogram of familiar sound. It is a tool that can be used to understand sound we can hear based on hearing threshold. Louder sounds toward the bottom of the graph, while softer sounds are towards the top. Low-pitched sounds are on the left, while higher-pitched sounds are on the right. Speech banana is a term used to describe the area where most sound of average conversational speech occur on this graph. While many other sounds fall outside speech banana, audiologists are most concerned with the frequencies within this speech banana, because hearing loss in those frequencies can affect a child's ability to learn language. Next, we will learn about the structures of ear. Ear is divided into external, middle, and internal ear. External ear is filled with air. It has pinna, which functions as diffract and focuses the sound waves, collects sound, and funnel it to the tympanic membrane. Cerumen, which lining the external auditory canal, which has antibacterial properties, protects from bugs, mold, foreign particles. We have middle ear, which also filled with air, which consists of tympanic membrane and ossicle chains. It is involved in impedance matching, which is a process in the transduction of sound. So the middle ear transfers the incoming vibration from the comparatively large low impedance tympanic membrane to the much smaller high impedance upper window. So there are some processes that are involved in impedance matching. First, we have hydraulic action of tympanic membrane. So since the surface area of tympanic membrane is much larger than area of stapes foot plate by the ratio of 21:1, the forces collected over the tympanic membrane are concentrated over a smaller area, thus increasing the pressure over the upper window. We also have lever action of ossicles. Since the handle of malleus is 1.3 times longer than the long process of incus, it provides mechanical advantage of 1.3. Tympanic membrane also have buckling action, since the force is transmitted from the center of the tympanic membrane, since tympanic membrane does not move as a plate. Lastly, we have inner ear, which is filled with fluid. It consists of bony labyrinth and membranous labyrinth. Bony labyrinth is the three semicircular canal, which is lateral, posterior, and superior, while membranous labyrinth contains ducts. We have three ducts: scala vestibule, scala tympani, and scala media. The cochlea and vestibule are formed from the bony and membranous labyrinth both. So what is the cochlea? This is the cross-sectional picture of the cochlea. It is a spiral-shaped structure that composed of three tubular canals or ducts and contains the organ of Corti. What is the organ of Corti? It contains the receptor cells and it is the site of auditory transduction. So the duct contains fluid, and the fluid in each duct has different composition. So in this table, we can see the differences between endolymph and perilymph. So endolymph is the fluid in the membranous labyrinth of the inner ear, while perilymph is the fluid between the membranous labyrinth of the ear and the bone which encloses it. Next, endolymph presents inside the membranous labyrinth, while perilymph surrounds the endolymph located inside the bony labyrinth. Endolymphs occur inside the cochlear duct, while perilymph fluid found in the tympanic duct and vestibular duct of the cochlea. Endolymph rich in potassium ions and resemble the intracellular fluid, while perilymph is rich in both sodium and chloride ion and resembles the extracellular fluid. Endolymph has more positive electric potential, while perilymph has less positive electric potential. Endolymph transmits sound wave to hair cell, while perilymph transmits sound wave to endolymph. Next, we will learn about the auditory transduction. It is a transformation of sound waves or sound pressure into electrical energy. So before transduction can occur, sound wave traveling through air must be converted into pressure waves in fluid. So the acoustic impedance of fluid is much greater than of air. The combination of the tympanic membrane and ossicles serve as impedance matching device that makes this conversion. So impedance matching is accomplished by the ratio large surface of tympanic membrane to the small surface area of the upper window, and mechanical advantage offered by the lever system of the ossicles, as we already learned earlier. So external ear directs sound wave into the auditory canal, as you can see in this picture, which transmit the sound wave into the tympanic membrane. So the tympanic membrane will move, the chain of ossicle also moves, and punching the footplate of the stapes into the upper window, displacing the fluid in the inner ear. Okay. Next, the organ of Corti that lies on the basilar membrane. Here's a picture of the organ of the cochlea, and this is the organ of Corti. So the organ of Corti lies on the basilar membrane of the cochlea and is based in the endolymph contained in the scala media. So auditory hair cells in the organ of Corti are the sites of the auditory transduction. Organ of Corti contains of two types of receptor cells: inner hair cells and also outer hair cells. Inner hair cells is fewer, arranged in a single row, while outer hair cells more numerous and arranged in parallel rows. Cilia protruding from the hair cells are embedded in the tectorial membrane. Thus, the bodies of the hair cells are in contact with the basilar membrane, while the cilia of the hair cells are in contact with the tectorial membrane. So the nerves that serve the organ of corti are contained in the vestibular cochlear nerve. Cochlear nerve is cranial nerve 8. So the cell bodies of the nerve are located in the spiral ganglia, and the axon synapse at the base of the hair cells. These nerves will transmit information from the auditory hair cells to the central nervous system. Next, we'll learn about the steps of the auditory transduction. So, auditory transduction by hair cells on the organ of corti occurs in the following step. Firstly, the sound waves are transmitted to the inner ear and cause vibration of the organ of corti. Auditory hair cells are mechanoreceptor, which are located in the organ of corti. Basilar membrane is more elastic than tectorial membrane. So this is the basilar membrane. This is the tectorial membrane. So this basilar membrane is elastic, so it moves with vibration. Vibration of organ of corti causes bending of the cilia on the hair cell by shearing forces as the cilia push against the tectorial membrane, as you can see in this picture. So bending of the cilia produces a change in potassium conductance of the hair cell membrane. Bending in one direction produces an increase in potassium conductance and hyperpolarization. Bending in other direction produces decrease in potassium conductance and depolarization. So these changes in membrane potential are the receptor potential of auditory hair cells. The oscillating receptor potential is called cochlear microphonic potential. So when hair cells are depolarized, the polarization opens voltage-gated calcium channels in the presynaptic terminals of the hair cells. Calcium enters the presynaptic terminals and causes release of glutamate, which functions here as excitatory neurotransmitter. Action potentials in the afferent cochlear nerves that will transmit this information to the central nervous system. When the hair cells are hyperpolarized, the opposite events occur and there is decreased release of glutamate. Oscillating depolarizing and hyperpolarizing receptor potentials in the hair cells cause intermittent release of glutamate, which produce intermittent firing of afferent cochlear nerves. So this is the simplified version of the mechanism of auditory transduction from what I have explained in detail earlier. So from sound wave, cause vibration of the organ of corti, bending of the cilia of the hair cells, then will cause the depolarization, which means change in potassium conductance of the hair cell membrane, and this process of oscillating receptor potential will cause intermittent glutamate release, which then cause intermittent action potentials in afferent cochlear nerve. Next, we will learn about the encoding of sound after the auditory transduction process. Encoding of sound happens. So encoding of sound frequencies occur because different auditory hair cells are activated by different frequencies. The frequencies that activate a particular hair cell depends on the position of that hair cell along the basal membrane. So the base of the basilar membrane is nearest the stapes, narrow and stiff. It responds best to higher frequency, while the apex of basilar membrane is wide and compliant. It responds best to low frequency. Basilar membrane acts as sound frequency analyzer, with hair cell positioned along the basilar membrane responding to different frequency. This spatial mapping of frequency generates tonotomic map, which then is transmitted to higher level of the auditory system. Next, I will explain a little bit about the traveling wave theory. It is a theory that introduced by von Beckersey in 1960. He said that sound travels along the basilar membrane in a series of waves, and that these waves peak at different places of the membrane. He said that low frequency waves peak towards end of the cochlea, while high frequency wave peak near at the entrance or base of the cochlea. Next, after the vestibular cochlear nerve pick up the action potential via its afferent, it will bring the information via the auditory pathway. So auditory pathway started at the 8th cranial nerve, and then we go to the ventrocochlear nucleus, and then we go to dorsal cochlear nucleus, lateral meniscus, inferior colliculus, and middle nuclear body, and towards the auditory cortex at the temporal lobe. The tonatomic map that was generated at the level of organ of Corti is preserved at all levels of central nervous system. Complex feature discrimination, like the ability to recognize a pattern sequence, is the property of the auditory cortex. But because some auditory fibers are crossed and some are uncrossed, a mixture of ascending nerve fibers represent both ears at all levels of central nervous system. Lesions of cochlea at one ear will cause ipsilateral deafness. However, most central unilateral lesions do not cause deafness because some of the fibers transmitting information from that ear have already crossed to the undamaged side. Next, auditory cortex. We have primary auditory area and associated auditory area. A primary auditory area number 41 and 42. It is the main receiving area of heard sounds. It receives neuron from medial geniculate body by auditory radiation. And lesion of bilateral temporal cortex causes cortical deafness. Associated auditory cortex, area number 22. It receives auditory impulses from primary area, and this area is responsible for the interpretation of the sound. So lesion of area 22 will cause word deafness, inability to interpret the meaning of heard sounds. Next, I will explain a little bit about acoustic reflex. It is also known as stapedial reflex. The contraction of the stapedius muscle elicited by the presentation of an acoustically loud sound. So when either ear is presented with a loud sound, the stapedius muscle on both sides will contract. The pure tone intensity range to elicit this reflex is 70 to 100 decibel. Contraction of the stapedius muscle tilts the anterior stapes away from the oval window and stiffens the ossicular chain. This results in increased impedance, which is measured as a small decrease in compliance by an ear canal probe. Stapedius muscle is innervated by 7th cranial nerve, which is the facial nerve. So in the presence of facial nerve paralysis, the stapedius muscle is likely to be affected. Thank you.